Thank you very much for the introduction and the opportunity to speak. So I'm going to take for granted that we're going to be hearing quite a bit more about the transformative potential of data and models to improve bioprocess performance, particularly Wednesday and Thursday. But today I'd like to make the case that models can be extended much further upstream in the sense of biological causality to genetic design. And applying models to genetic design really allows us to approach this question of, can we holistically optimize the genetic design and the bioprocess design to achieve much better performance in our systems than by individually optimizing each of those? So this motivation kind of follows my own professional journey. I started modeling bioprocesses by doing computational fluid dynamics of bioreactors, right? Looking at optimizing oxygen transport. And fairly successful in doing that, and then started asking, well, what about the cells, right? If we want to know about the performance of the bioreactor, we should probably consider the cell's behavior. And about the time I joined Asimov, I had the opportunity to ask, well, what about the cells, right? What about their genetic design modulates and allows us to achieve higher bioprocess performance? So for those of you who are not familiar with Asimov, our mission is to bring the computer-aided design revolution to biotechnology. So in much the same way that electrical engineering industry was transformed by CAD tools that brought automation and design rule checks, performance prediction and design optimization, we're building the first industry-grade computer-aided design platform for our industry. And to give you an idea how that concept of CAD for biology fits into our platform. So we have a suite of really hyperfunctional genetic designs and host cells that we can tune using our CAD software, which we call kernel, and our models in order to produce very high-producing, high-performing cell lines that we tech out to our clients to express their molecules. And then, of course, we can use our models and our software internally to improve the quality and the performance of our services as well. So our vision is really to transform synthetic biology and bioprocess design into a mature engineering discipline, engineering being the key here, right? That uses digital technologies to multiply the benefit of our industry on human health. So I think that's really realized in its ultimate form by this idea of holistic genetic and bioprocess design. And to understand what I mean by holistic genetic and bioprocess design, I think it's helpful to contrast that with the current method by which we do bioprocess development, which is represented here on the top. It's a very empirical process, right? We clone our vector, we transfect that into our cell line, we select the optimum cell based on that genetic design, and then we move into upstream process development, right? Media selection based on now a fixed top cell line, and then process design based on a media and a cell line. So contrast that with the digital development process, where we're able to model all of these things in silico: the genetic design, the cell line optimization, perform media optimization in silico, and do process optimization in silico before we move into process qualification. Well, obviously, we can now replace that iterative, empirical, costly design process, but we can also optimize all of those things not independently, but mutually, right? And so if we're optimizing the choice of our host cell line, the choice of our genetic design, at the same time we are optimizing the bioprocess, then we can achieve much higher performance than if we were to optimize each of those things independently. It also allows you to do things like intrinsically account for quality by design, right? By including in your objective functions of your optimization certain critical to quality attributes. And of course, it has all the in silico benefits of reducing attrition and rework, and then increasing time to market, or decreasing time to market, increasing speed to market. And we're pretty well positioned as a modeling and simulation team at Asimov to realize this goal of holistic optimal design. And that's because our modeling and simulation capabilities are really well supported by our empirical capabilities. So we have very strong synthetic biology and cell line development and process development teams that can create these genetic designs, assemble the DNA, measure genetic part attributes, perform multi-omics analysis, and then in bioprocess development, right, measure all of the omics and do all the experiments to confirm performance. And all of these things generate data that we can use in our models and then feed back into the design. And so that gives us the ability to make these end-to-end representations from gene expression to phenotypic variants, to metabolism, to bioreactor transport that then allow us to maximize growth or productivity or quality. So it's also worth mentioning that this concept of end-to-end upstream modeling is applicable across modalities. I'm going to focus on CHO today, but we've actually launched multiple platforms. We've launched our CHO Edge platform expressing antibodies in CHO and our lentiviral expression system in HEC293 cells. And we're on the verge of launching an AAV system also in HEC293 and currently working on an RNA manufacturing platform as well. So I'm going to describe a few applications of models that apply across this entire portfolio, and then one particular application to CHO specifically. So this is going to be the roadmap for the rest of the talk. So all of the phenomena that we model and I'm going to talk about today are represented here. We can think of this as central dogma plus, right? So the idea here is that we have our exogenous genetic design, right? Either transiently transfected or stably integrated. That's transcribed into mRNA, translocated out into the cytosol, where it's then translated by ribosomes, codon by codon, into our growing protein chain. And of course, at the beginning of that protein chain is often a signal peptide, which helps chaperone that protein into the secretory pathway, into the endoplasmic reticulum, where all of the post-translational modifications take place. And so that signal peptide ends up being important as well. Now, of course, we're not only concerned with secretion and the expression, right, but we're also concerned with metabolism because it both supports and competes with expression of our gene of interest. And we're concerned with the proliferation of the cells in culture because that scales but also competes with expression of our gene of interest and with the titer that we ultimately achieve. So first, I'm going to zoom into just the effect of genetic design on translation. So one fundamental constraint that we often operate under, although we always operate under practically, is that after discovery and lead optimization, we have a fixed amino acid sequence. So we can't optimize our amino acid sequence to optimize performance. But what we can optimize is the codon sequence that encodes that amino acid. So our codon optimizer is an algorithm based on a model of natural codon usage that adapts the codons for a given amino acid sequence to optimize the usage that would support translation, but also to minimize the inclusion of unintended, deleterious motifs, right, that might be introduced in the nucleotide sequence. So what we do is we start with the relative codon usage of each codon expressed in some representative selection of the relevant genome. And then we can use a dynamic programming algorithm to select the best codon sequence for a given amino acid sequence, excluding then those problematic motifs that might interfere with translation or any other effects. And when we input an unoptimized amino acid sequence, our codon optimizer will produce the optimal codon sequence that maximizes the similarity of that codon sequence with our host genome in a couple of key ways. And when we use those codon optimized sequences to express antibodies in our CHO platform, we see titer increases that average about a 70% improvement and up to a threefold improvement over not an unoptimized sequence, but over the second best third-party codon optimizer, right? So huge improvements in the CHO expression. But also interestingly, we also see dramatic improvement in lentivirus expression in our HEC293 system. And in that case, the functional titer will increase 12-fold on average, 17-fold in some extreme cases over an unoptimized sequence in this case. So really huge benefits from a very simple model, but key is the right data set here. Now, continuing post-translation, we find the role of the signal peptide. So I described this, right? It's recognized as chaperoned into the ER, and it destines the protein for the secretory pathway, but miscleavage of that signal peptide or lack of recognition of that signal peptide can lead to yield loss, right? It can lead to cell toxicity if the protein products are not actually chaperoned to the ER and have various deleterious effects on titer ultimately. So it's critical to efficient secretion of that protein. And some signal peptides are going to be more effective than others. So we developed what we call the signal peptide cleavage predictor. It's based on an existing model in the literature, but we've added some novelty to it. And what it does is it not only recognizes the type of the signal peptide, but it predicts the location and probability of cleavage sites for that combination of signal peptide and CDS. As I said, it's improvement over the published state-of-the-art, which is a model called Signal P 6.0. And what we do is we have our own more elaborate prediction head that we add to a protein language model that is fairly common. It's called ProtBERT. That's a large language model for protein embeddings. And after we train that on public data, we're able to actually predict with a high competitive and actually exceeding the state of the art a little bit, the precision and recall of the prediction of the cleavage site and the probability of cleavage at that site. So more interesting than the model itself in this case is how we use it. So in this particular case, we found a little bit over 3,000 potential signal peptides from public databases. And we combined those fully combinatorially with 200 biologic sequences to get about 600,000, a little over 600,000 potential signal peptide CDS sequences. Then we put these into our signal peptide prediction algorithm. And we were able to select 1% of those that had consistently high cleavage probability and consistent site that we thought, okay, these might be candidates, right? They cleave in the right site. We think that they're going to effectively destine this this protein to the secretory pathway. Now let's test them for performance. And when we did test those for performance in stable pools, we saw the top of those cases, this new signal peptide 8, yielding a 62% increase in our antibody pool titer over kind of the industry standard signal peptides that we had been using. Now, zooming back out to central dogma as a whole, we find our genetic circuit simulator, which is one of the first models developed at Asimov. And if you'll forgive a little bit of a pun, is one of Asimov's foundational models, right? So in this case, it's a mechanistic model of expression dynamics that starts with genetic design, and then it combines logical and algebraic representations of cis and trans regulatory effects with kinetic descriptions of transcription and translation, and of course, closure and mass balance to be able to predict the dynamics of expression of our genes of interest. So transcription is kind of represented here on the bottom left. It's basically an accounting of initiation and termination. So initiation is typically scaled by transregulatory effects. So the promoter strength or the activity of a particular promoter in the presence of transcription factors of a particular abundance, right? And then propagation of RNA polymerase or of ribosomes often is scaled by cis-regulatory effects, right? So how effective is your terminator? How leaky is your terminator? So as an example, we have a genetic circuit here, which is 3 transcription units, right? So 3 combinations of a promoter, a CDS, and then a terminator. And what our genetic simulator will do is march along that genetic circuit and then predict the rate of expression, the rate of transcription, and achieve predictions of the abundance of mRNA transcripts at a given time. So for 3 transcription units in series, you could have as many as 6 possible mRNA transcripts, right, associated with either, perfect initiation and perfect termination, or read through the first terminator and then termination on the second terminator, right, initiation on the second promoter, termination on the second terminator, right, and so on and so forth. So there's 6 combinations, and we can actually predict the levels of expression of those at a given time. And so that's kind of the source term in our model. The rest of the model of translation, of complexation, secretion, decay is basically reaction models, right? So a set of ordinary differential equations, nonlinear ordinary differential equations that we build to represent each of those reactions. And then by integrating that over time, we get the dynamics of expression. So we've previously validated this genetic circuit simulator on fluorescent proteins, and we're currently in the process of validating it on antibodies, both canonical mAbs and bispecifics with multiple architectures. But even before we've fully validated this quantitatively, our synthetic biologists have found this useful. And so we've deployed this internally in our kernel CAD software for our synthetic biologists to use. So a typical use case might be motivated by this observation that certain mAbs, right, are best expressed by certain combinations or certain ratios of heavy chain and light chain expression. And a different mAb will have a different preferred heavy chain, light chain ratio of expression. And so a use case might be that we go in and we select a slate of plasmid designs such that we are spanning a dynamic range of heavy chain light chain ratio intentionally, right? So we'll use the simulator to design those plasmids that span that range of heavy chain light chain ratios. Then we'll synthesize those, combine those with our mAbs, and then we can select the design that best expresses or that achieves the best endpoint titer for that particular mAb. Of course, to get from genetic design to process performance, we of course need to consider the cell population and its behavior in cell culture. And particularly, how does metabolism support and compete with antibody expression, as I said, and how does cell proliferation scale and compete with titer? So what I'm going to show you now is our CHO simulator. It's a suite of machine learning, hybrid, and mechanistic models that describes all of those phenomena. So in one particular instantiation, I'm going to focus on the hybrid instantiation of our CHO simulator here. It's composed at its core of a metabolic flux analysis or a flux balance analysis, which represents the cell metabolic network. And it makes an assumption that the cell doesn't accumulate anything as part of its metabolism, right? So basically mass is conserved within the cell. But this is typically, well, in typical situations, this would be combined with a set of ordinary differential equations that represent mass balance extracellularly, and we would call that dynamic flux balance analysis. Our implementation is a little bit different. It's modulated, our fluxes are modulated by an artificial neural network that we have trained to recapitulate the data that we see in various process conditions. But what we're doing is we're representing 40 dynamic states, 32 of which of those are extracellular metabolites, one of which is a mAb, and then 3 unique cell states and 5 bioreactor states that represent things like pH. And we integrate that over time and we can get trajectories of all of those states. And then, of course, we can do things like optimize. So again, a typical use case might be something that we represent here, where this is how we train the model, right? So we have a set of experimental conditions that represent a fairly wide variety of process conditions, feed strategies, for instance, media compositions, and different mAbs. I'm showing you here 8 different metabolites on the top 2 rows. So the first one is actually not a metabolite, that's viable cell density. And then we have glucose, and I believe lactate, ammonium, pyruvate, alanine, glutamine, pH, I think, represented here. And then on the bottom, 4 different mAbs. And you can see that as we train the model to learn its parameters, it will begin to recapitulate the experimental data quite faithfully. So now that we've trained the model, we can start to apply it. And so a particular use case might be, first, we predict the trajectory of some baseline process, let's say our platform process, and then we can of course confirm that the predicted trajectory of that process matches our current process. But then we can run an optimization problem and say, given certain decision variables for this process, let's say feed quantities, let's fix the feed times, but let's just let the model decide the feed quantities to provide to this process. With an objective of maximizing titer. So titer here is represented on the bottom right panel, and you can see the green trajectory is the result of allowing our optimization to maximize titer, right? Maximize endpoint titer specifically. And so what we do see after experimentally validating this with titer optimization is that in one particular case, one mAb, we saw a 20% increase in titer, and in another mAb, using a range of different conditions that the model predicted, we saw an average of about a 7% increase in titer just through application. So not only can we use this CHO simulator model to optimize processes for individual products, but we can also optimize our platform generally. And so that's what I'm going to show here. So we've applied the CHO simulator to improve our platform clone selection process. In our incumbent process, what we call the CHO Edge Platform 1.0, we will typically expand 144 clones, and we'll assay those at increasing scales. But then we'll screen those and we'll tape just the top 46 clones based on just the final titer at that last stage of expansion. And we will advance those to our clone selection process, which is an AMBER 15 fed batch process. And we'll do that, advance those 46 clones with just one platform feed. So that's what we'll be using on the AMBER 15 is just 46 clones by one feed strategy. So using the CHO simulator with an appropriately defined objective function, we were able to first reduce the number of clones that we advanced through an ancillary model, a machine learning model that just used that small scale data in a little bit more intelligent way to screen for the top 16 as opposed to the top 46 clones. But then we use this CHO simulator to select platform feed conditions, 3 platform feed conditions that we expect to benefit from the diversity of those clones that we advance. So some feed strategies might be preferable to a clone expressing one molecule. Another feed strategy might be preferable for a clone expressing a second molecule. And by screening these now 16 clones on 3 different feed strategies, we expect to find the best combination of clone and feed strategy for that particular molecule. And we do see marked titer improvements using these models that embrace clonal and molecular diversity. So we see anywhere between 4 and 38% titer improvement in our 14-day fed batch process. And you can see, as an example, we have 2 candidate feed strategies that were selected by our model. We'll call them feed strategy A and feed strategy B. They're VCD-based feed strategies, and so there's a little bit of variability depending on how those cells grow in the actual feed. But you can see for our two medias that we feed, one of these, let's say both of them actually will predict that we want to feed a little bit less in the early exponential phase, support growth and stationary phase pretty well by overfeeding a little bit, but then they diverge in their strategy in the decline phase, where one feed strategy looks like it wants to be fairly close to our baseline, and the other strategy wants to feed much less in the decline phase. And you can see that the cumulative feeds, we tend to have one feed strategy that favors a slightly higher overall feed, and another one that favors a fairly low overall feed concentration. And what we'll see is when we screen molecules, a panel of clones, each expressing a different molecule on these 2 different feed strategies. For one feed strategy, we might find 4 out of the 5 molecules do better than our platform strategy using that feed strategy. But we have one case here, right, that didn't like that feed strategy. In our second feed strategy, we have one molecule that really likes this new feed strategy. The molecule that didn't like feed strategy A, it still now outperforms in this second strategy relative to our platform. 2 different molecules underperform in that feed strategy. Now, you might think, well, okay, that's lucky, right? That you happen to select 2 that are complementary, but our objective function, remember, was actually to embrace that diversity. It was defined in such a way that we are selecting for diverse feed strategies that will prefer different clonal phenotypes, or that different clonal phenotypes will prefer. And we do see when combining those and taking, the best performance out of feed A or feed B, we see an average of about 15% improvement. So can we model the effect of genetic design on fed batch performance, on bioprocess performance, and on titer? I would say emphatically yes. And by applying those models sequentially, what we've achieved is about a 3.5-fold improvement in our average antibody titer and about a 12-fold average improvement in our lentiviral titers. And now we're achieving routinely 6 to 11 grams per liter clonal antibody titers in our CHO platform, up to 1E9 TU per mil in our stable producing clones for our lentiviral system, and about 1E8 TU per mil in our packaging cell lines for our lentiviral system. So can we optimize things holistically? I would say we have now laid the groundwork, and to stay tuned for the actual holistic optimization. And with that, I will stop and take questions.
Okay, thank you very much for a very interesting presentation. Take some questions. Hi, thank you for your talk. Kind of talking about the data that you have at the end about your packaging cell lines, and I guess kind of leading into the data that you're going to have with AAVs. How exactly do you go through modeling, say, like the transient transfection process, where there's a lot of key attributes about that process that's going to impact your titer that may, vary with scales and things like that?
That's a very good question. I'll take a philosophical take at that first, right? Which is the more established a process is, the more it admits complex model application, because you have tighter margins, you're trying to improve, much take smaller steps, right, on a more fixed process. For something like a new platform that's in development, we can actually apply much simpler models. So to directly answer your question, a lot of the models that we apply to, let's say, our lentiviral expression platform in development were statistical models, right? We're looking at specifically to the question of transfection process, minimizing the uncertainty in that process, right? Identifying the sources of uncertainty and then mitigating them. To be a little bit more forward-looking, once that process has become stable, particularly for transfection process, we could start looking at mass transport models of the transfection mix itself, right? Ensuring that we have adequate mixing and that we have, the correct dose, basically, of, your transfection reagents and your DNA reaching the cells. And then we could also start looking, and I think there's great opportunity here, and I'm really itching to try it out. How does the gene expression dynamics of your different components, right, in a viral vector expression system, how do the rates of expression of each of those then influence your overall titer as it relates to, growth, but also your quality attributes, right, like your percent full in an AAV, right? You could imagine that the dynamics of expression of those things are very key. Yep.
Thank you. I just had one more question, if that's okay. The other thing I was curious, and this may depend on the platform or the molecule that you're trying to produce, but overall, for your training set data, how much does it really take for your machine learning algorithms to converge on the right values of your parameters?
So it depends on the model, but I'm inferring that you're referring to the CHO simulator model where we have the fancy animations. Yeah. So very little, in fact. So we can train that based on just one process, one 14-day fed batch process. What we need is the metabolites that are part of the model to be measured, and we need those that are relatively high frequency. So daily measurements is typically sufficient. Everything else, so that's sufficient to train it to really recapitulate one process, right? Or, a platform process. Where I think we start to really see value is in training that on a variety of processes, and the neural network component learning what is it about, the different conditions in those processes that modulates the metabolism. And so for that, what I showed you, I think we're training that on 40 different processes, and it does a fairly good job of extrapolating to conditions it hasn't seen.
Thank you.
Yeah, pleasure.
And thank you for a very nice talk with the combination of all the modeling in silico and the feed to the end of the upstream. So a little bit on the how you optimize the feed strategy. Is that based on came from the model and you go with the platform design of feeding higher in the growth phase and flat down later, or like you look on the VCD or some other the metabolic parameters, like what you work on, like how you work a little bit on that side?
Yeah, so in this particular case, we went in with an IVCC based feed, right? So the feed is based on two parameters on any given day, the slope and the bias of this IVCC based feed. And that's something that's commonly implemented in fed batch processes. So that's not model related. So what we told the model is those slope and bias parameters. Tell me the slope and bias that are optimal for any particular viable cell density that you're going to see over the course of a process run. And it selected blocks. We did constrain it to say, for day 0 through 3, we want a particular slope, and then for day 3 through 6 we want a different slope, but then we allowed it to choose those levels. Hopefully that answers your question. Yep. Mm-hmm.
Hi, thank you very much. That was a really cool talk.
Thanks.
For the CHO simulator, the work of applying it to cell line screening is awesome. it seemed like you went from the 144 clones to 16 clones using your machine learning model. When you did that, were you able to get all the metabolite information you needed from the plate-based 144 clone screen, or did you have to do additional screens in that workflow and extend the time of your?
So no time extension. This does open up a bit of a can of worms, but short story is we didn't use any metabolomics data for those particular clones to select the feed strategies for those clones. The feed strategies were selected based on training on historical data at that point. So we don't need to fine tune the model for individual clones. What I'm working on at the moment, I'm very excited about, is the ability to call it zero shot, predict the fed batch titer and optimize the fed batch titer based only on the sequence of the incoming molecule, right? So that's the exciting evolution of that. Yep.
Yeah, that's awesome. Thanks.
Okay, maybe one final question.
Excellent talk. Thank you so much. So I have a question based on the incoming sequence of like a novel molecule. Like in my experience, I found that like every time we try something in the lab, it works for one molecule, then we move it to the next molecule, and then doesn't work as great. How are you navigating that, and how much like input do you actually need to like make a good, reliable prediction?
Yep. So I will say what's in the model right now is we know the stoichiometry of that molecule. We know its amino acid composition, and of course the ATP requirements to express that particular composition of amino acids. What I alluded to that we're working on that is still very much in development is AI or machine learning models that will take the actual sequence and make predictions as to the gain that we expect to see in titer for that sequence. And then, of course, when we incorporate that with the metabolism, we know, okay, how much extra burden is that going to put on the cell, right? So hopefully that answers that question. Yep.
Okay, thank you very much to Will for a really interesting presentation. So thank you very much.